3, 2, 1. The amino acid sequence of a polypeptide is programmed by a unit of inheritance that we call the gene. And these genes that make up our genome, that make up our genetic information, are made of DNA, deoxyribonucleic acid. So these genes are a nucleic acid. In fact, they are a polymer of nucleic acids. There are two types of nucleic acids, ribonucleic acid and deoxyribonucleic acid. So deoxyribonucleic acid has an extra oxygen, if you will, compared to ribonucleic acid. They're both 5-carbon. They are both 5-carbon nucleic acids. DNA provides directions for its own replication. RNA synthesis is directed by DNA. In fact, DNA through messenger RNA synthesis controls protein synthesis. Protein synthesis, of course, occurs in the ribosomes. Your DNA is inherited directly from your parents. Each DNA molecule is very long and consists of hundreds of thousands of hundreds to thousands of genes. Before a cell can reproduce itself, it has to copy its DNA. It has to make a copy of its DNA. The copies then are passed on to the next generation of cells. While DNA does encode the information that programs all the cell's activities, it is not directly involved in the day-to-day operation of the cell. Those instructions that are directed by the DNA that operate the cell are actually contained in the proteins that are created by the DNA, excuse me. Each gene along a DNA molecule directs the synthesis of a particular type of messenger RNA molecule, which in turn directs the synthesis of a particular protein in the ribosome. So the flow of genetic information is from DNA to RNA to protein. This is known as the central dogma of molecular biology. Again, it's the central dogma of molecular biology, DNA to RNA to protein. In eukaryotes, the DNA is located in the nucleus, but most ribosomes are in the cytoplasm. So messenger RNA functions as an intermediary, moving the information from the nucleus, from the DNA, to the ribosomes to make the protein inside of the cytoplasm. Prokaryotes, on the other hand, lack nuclei, but still use RNA as an intermediary to carry a message from DNA to the ribosomes. We'll talk about the evolutionary importance of that a little bit later on. So graphically, what we're looking at here is the DNA here contained in the nucleus, where it synthesizes messenger RNA inside the nucleus, and then the messenger RNA is excreted or exported from the nucleus to the cytoplasm via nuclear pores. These nuclear pores are 100 angstrom openings inside the nuclear membrane that it's the perfect fit to allow messenger RNA from the nucleus into the cytoplasm. Once in the cytoplasm, the messenger RNA will interact with the ribosome to bring together the amino acids that match up to the coded message on the messenger RNA to produce a polypeptide. Now, nucleic acids or polymers, nucleic acid polymers are called polynucleotides. And I've said a few times that physiologists, biochemists, and molecular biologists tend to recognize polynucleotides up until it's about 100, that's wrong. Polynucleotides are polymers of nucleic acids. And each of the polynucleotides is made up of each of the polynucleotides are made up of individual nucleotides. Each nucleotide consists of 3 pairs or 3 parts: a nitrogenous base or a nitrogen-containing base, a pentose sugar, a 5-carbon sugar. In particular, we call it a ribose sugar, and a phosphate group. Phosphate group, again, is a phosphorus with 4 oxygens attached to it. The nucleotide without the phosphate group is called a nucleoside. So, here we have a nucleotide. Located on the nucleotide is the phosphate group, this guy right here, PO4. Notice that it is negatively charged. The nucleoside consists of the sugar, the pentose sugar, 5-carbon sugar, and the nitrogenous base. Trying to get my eraser to work here. Not working today. Okay, now, the nitrogen bases are rings of carbon and nitrogen that come in 2 different types, purines and pyrimidines. The purines, I want you to recognize the purines are adenine and guanine, so AG, and I remember it by saying that it is an agricultural product because Purina makes agricultural products. So I remember A and G, adenine and guanine, are the purines, and the others are pyrimidines. The pyrimidines are cytosine, thymine, and thymine is only found in DNA, whereas uracil, the final pyrimidine, is only found in RNA. So again, thymine only in DNA, and uracil only in RNA. Pay attention to a couple of things here. Pyrimidines a big name, but pyrimidines have 5 carbon rings. Purines are little names, but they have a 5 carbon ring and a 6 carbon ring. I'm not going to ask you to remember structure other than that, recognizing the number of carbons. The sugars now, let's talk about the sugars. In RNA, it is a ribose sugar. So here we number the carbons from the oxygen chain, from the oxygen atom, 1, 2, 3, 4, and the 5th carbon, the 5' carbon, is outside of the ring. On the number 2 carbon here in RNA, in the ribose sugar, we have a hydroxyl group. Same thing here for the number 5 carbon. We've got our 5 carbon not part of the ring, but on the new carbon, the number 2 carbon here, you will notice that there is no oxygen, hence the name deoxyribose. So the only difference between the sugars is the lack of an oxygen atom on carbon number 2. Okay, now because the atoms in the nitrogenous bases and in the sugars are numbered, the sugar atoms have a prime after the number to distinguish them. So the reason we have 1 prime, 2 prime is to tell us that that's the carbon on the sugar as opposed to the carbon on the nitrogenous base. So the combination of pentose and a nitrogenous base is the nucleoside. Add that phosphate group to it, it becomes a nucleotide. Okay, again, in written out form here, I've got what the purines are and the pyrimidines. Remember, it always helps me to remember purine, purina, A and G, adenine and guanine. Okay? Sugar, of course, in DNA is deoxyribose. In RNA, it's ribose, missing that oxygen on the number 2 prime carbon. And notice again, just to reiterate, just to make sure we don't have we don't forget, the addition of the phosphate group makes a nucleoside a nucleotide. And more correctly, we don't call it a nucleoside, we call it a nucleoside monophosphate. I'm sorry, a nucleotide monophosphate. So if you hear me say that, it's the same thing as a nucleotide. Now, polynucleotides are synthesized when adjacent nucleotides are joined by covalent bonds. So we'll have a covalent bond that forms between the hydroxyl group on the 3 prime carbon of one nucleotide and the 5 prime carbon on the next nucleotide. This will then create a repeating backbone of sugar phosphate units, with the appendages being the add-on being the nitrogenous base. Notice everything's the same. The only difference would be what nitrogenous base we have attached to that sugar. And the sequence of those nitrogenous bases, are unique for each gene. So the difference in genes is the sequence of the nitrogenous bases. It's also important that we recognize that the 2 free ends of the polymer are distinct. One end of the polymer has a phosphate attached to the 5 prime carbon. This is the 5 prime end. Let's go back here and take a look at that. Here we have a 5 prime end where we have the phosphate group attached to the end of the nucleotide. We call this the 5 prime end. The other end, we have the hydroxide group attached to the 3 prime carbon, and so we call it the 3 prime end. And this is important because we can only add bases in one particular direction. When we start copying the DNA molecule, there's a directionality to how it can be copied. Because genes are normally hundreds to thousands of nucleotides long, the number of possible base combinations and sequences is virtually limitless. And so the linear order of these nucleotides specifies the order of the amino acids, which of course, if you might remember from yesterday, is the primary structure of the protein. And the primary structure determines the 3-dimensional structure, which determines the function. Now, Let's go back here. Polynucleotide, talked about that. Adjacent nucleotides are joined by covalent bonds. I talked about that. These create a backbone of sugar phosphate units with nitrogenous bases as appendages. The sequence of bases along a DNA or messenger RNA polymer is unique for each gene. Okay. RNA, the RNA molecule, is a single polynucleotide chain. In contrast, DNA molecules have two polynucleotide strands that twist themselves around an imaginary axis to form what we call a double helix. So the double helix is a generalized shape of the DNA molecule. This double helix concept was first proposed in 1953 by James Watson and Francis Crick. We'll talk about them a bit more later. Francis C-R-I-C-K, Francis Crick. So the two backbones, the two sugar phosphate backbones, are on the outside of the helix, and they run in opposite directions. So remember, we talked about 5' 3'. They run in an anti-parallel fashion. So one strand has the 5' end here, which of course is where the phosphate and joins the other strand. And so the other strand will go from 3' to 5', so it's making this long backbone, right? And then the nitrogenous bases hold them together, kind of like a ladder. And then if you can imagine that ladder being twisted around an axis, that's the shape. That's what we call the helix, the double helix. So they run in opposite directions, and we call that anti-parallel. So they run in opposite 3', 5' directions in an arrangement that we refer to as anti-parallel. So pairs of nitrogenous bases, one from each strand, connect the chains together with hydrogen bonds, and I'll talk about that more when we talk about molecular biology. One DNA molecule has thousands to millions of base pairs. Now, there's lots of different genes, but there's thousands, hundreds of thousands to millions of base pairs on each of those DNA strands. Because of their shapes, only some bases are compatible with each other, and we recognize this, and we'll talk about this a little bit later on. We recognize that adenine will always bond with thymine, and cytosine will always bond to guanine. This is called Chargaff's CH. Oh, Lord. It's not what I wanted to do here. I'm going backwards? I think so. Yeah. This is Chargaff's rule. A-R-G-A-F. Chargaff's rule. I'll get better with this as time goes on, but that's all I got for now. All right? So here's a kind of a visual representation of what we've been talking about. Here you can see 5' and 3' in opposite ends. They run opposite in the anti-parallel direction, right? So the sugar phosphate backbone are these ribbons. And here you can see that the base pair using hydrogen bonds. So there are two strands, and the two strands wrap around a central axis. So prior to each cell division, we're going to have to make copies of the DNA. So one strand is going to serve as a template. So the blue strand here is the template. The blue strand is the template, which the orange strand or gold strand or whatever color that is the new piece of DNA that's being made for the daughter cells. So when I distribute the DNA to the daughter cells, I do so in a semi-conservative mechanism, so that the daughter cells will get one of the original parents' DNA strands and a new strand that's made as a result of synthesis. So what this does is it ensures that a full set of genetic information is transferred to every cell that's reproduced. So DNA and proteins can be used as tape measures of evolution. As an evolutionary biologist, this is huge. This is the new evolutionary investigation by biologists. So genes and the products of genes, proteins, document the hereditary background of your organism because it's passed from parents to offspring. So siblings have a greater similarity in their DNA and protein than do unrelated individuals of the same species. And so two more closely related species are more similar in their DNA than less closely related species. So this argument can be extended to develop a molecular genealogy, if you will, to relationships between species. So we can use this information to assess evolutionary kinship. Two species that appear closely related based on fossil and molecular evidence should also be more similar in DNA and protein sequences than are more distantly related species. So the close evolutionary history of primates will show more similarity in their DNA sequence than a primate would to maybe a bird or to maybe a reptile. So, for example, if you compare the sequences of 146 amino acids in a hemoglobin polypeptide, there are 146 of them, we find that humans and gorillas will differ in just one amino acid. That's just in their hemoglobin molecule, of course. That's only one particular chain of hemoglobin. More distantly related species than these two closely related primates would have more differences. And I should have put in the chart that showed some of that, but I'll get to that when we talk about molecular biology. So what I did write down, though, was that humans and gorillas vary only in one amino acid. In hemoglobin. HB is hemoglobin. Okay? If we compare humans and mice, the variation is 27 amino acids. If we compare humans and frogs, the difference is 67 amino acids. So here we have here we have a human, here we have two primates, comparing two primates. Here we compare a primate and another mammal. And here we compare a human and a reptile, amphibian. Make that human. And here we compare an amphibian and a human. So you can draw the inference that gorillas are more closely related to humans than are mice, which is more closely related to human than are frogs. So we often use molecular biology now to access evolutionary kinship. In fact, much more so than morphology anymore.